Hello, Eugene Aiello here. Thanks for joining me in the continuation of this series. In this set of videos, we're going to be discussing what happens when electromagnetic energy interacts with the skin. In the last video, we went into some detail about pigments, talking about how the pigment system absorbs electromagnetic energy and then transfers it. We're going to touch on where that transfer occurs, but we're going to more importantly discuss what's actually happening in the skin. So the first topic I want to start off with is when I was using cold lasers for multiple years, and most people in the orthopedic setting, they're being told that cold lasers are increasing, the red cold lasers are increasing mitochondrial function by interactions within the mitochondria. The biggest sticking point with that is that direct effect of piercing through the tissues and that red light interacting with cytochromes makes no sense. It's an impossibility. And what I mean by that, near-infrared lasers are going to be the only lasers that penetrate through this epidermis layer of skin. They'll penetrate as deep as the dermis, but your muscles and ligaments and tendons are below that subcutaneous level of subcutaneous fat. So even near-infrared lasers are not getting anywhere near the target organs. More importantly, red light is only going to penetrate into the epidermis, maybe partially through, not even hitting that basal membrane. As you go further down the spectrum, orange, yellow, green, blue, indigo, violet, violet is going to be the least amount of penetration. So as you go through that scale, the penetration changes. And when you get into further in the infrareds, it starts getting more complicated and less uniform because of the interactions that those have with water. So you kind of get a deep penetration, then all of a sudden it's very superficial, but not important for our topic. But either way, none of these different lasers are going to be penetrating as deep as muscles and ligaments and tendons. I find it fun when I have laser reps show up at my door and want to talk about their new greatest laser. And well, this laser is the deepest penetration. It's, so many watts of photonic energy, and it kind of drives me crazy because the misinformation when it comes to laser therapy. If you were to increase the intensity, the wattage of any of these lasers, it's not going to change the penetration depth. Penetration is dependent on wavelength. The reason that is photonic energy is going to interact with different tissues. So it's going to absorb into it. It's going to get transmitted, scattered. A lot of it's going to be reflected. So the physics behind it is very important, but it seems like in that field, they don't care about the physics. They're just trying to sell you a $40,000 laser. And then they have these features where they pulse so that laser beams going in and stopping, it's going at different frequencies with the idea of expanding and contracting the cells, which I don't even, besides near infrared having effects on water structuring, I'm not quite understanding where any of this comes from. So I like to get through what I call BS and get to what it's actually doing. I've had experience, as many orthopedic physicians have, using these red lights, lasers do have effect on the healing time of muscles. You do see that there is a benefit, but it's obviously not a direct benefit. The indirect effect is what this whole series is going to talk about. There is a biophotonic system. This photonic energy is getting absorbed into the skin, and somehow that's transferring into the system, and somehow that system is eventually helping tissues heal. Yeah, there's probably more, but we're going to discuss that later. Different elements of physiology are changing. It's not as simple. It's not as limiting as just saying that laser therapy is going to speed healing time. Laser therapy at different frequencies are going to have all kinds of different physiologic effects, which we'll discuss as we go through this series. But first, I want to talk about what is in the skin, what cells are in the skin. Our skin is primarily made up of keratinocytes, which is represented on these guys. And I know this isn't the most accurate drawing. Those keratinocytes are going to be intermingled between all these, and obviously the size is not matching with reality. So these keratinocytes will make up the majority of the skin. They start dividing at this basal membrane, so they're rapidly reproducing. I think it's 45 days that you turn over your skin. So they mature as they get further and further to the surface, eventually getting to this top layer where they die and they form that keratinized coat of the skin, which is a protectant, obviously. The reason they die is our blood vessels are only in the dermal layer. So again, when you're talking about this, you're going to get some blood interaction with the near infrared and possibly the red. but the others are not going to interact with blood vessels. So blood vessels are diffusing nutrients and oxygen and pulling carbon dioxide and toxins from those cells, no direct contact. That's why as they get further from that basal membrane, they die, they can't survive. So those are your keratinocytes. So we'll go back to them in a minute. The next cell is a fabulous cell. It's very interesting, and it's going to be a very large topic of an entire video. It's the Langerhans cells. Langerhans cells in conventional science are taught as white blood cells, the sentinels. Those are the ones that are monitoring the environment. They're found in your mucous membranes, they're found in your skin. They're also found in your liver, your spleen, a few other locations and organs. But classical view of the Langerhans cells as a white blood cell is they detect foreign invaders. And when they see a foreign invader, they engulf it, they process it, they produce an antigen that would be the protein that would distinguish that as something else. After they figure out what that antigen is, these Langerhans cells will then migrate to the lymph nodes. They'll meet the B cells there. They're going to expose that antigen to the B cell, which is going to become a plasma cell and producing lots of antibodies against whatever that antigen was. One of the sticking points with this is that Langerhans cells embryologically come from the same stem cell line as the skin and nervous system. Your white blood cells and your red blood cells come from mesodermal line, which these guys are ectodermal line. Mesodermal line, completely different stem cell lines, so they're not even embryologic related to the immune system. The other interesting part I found in my research is nerves are actually making synaptic-like contact with Langerhans cells. More importantly, the blue part of this is the glial cells. Glial cells wrap around every nerve in our body. Those glial cells make a synaptic contact. To make matters more interesting, embryologically, the Langerhans cells are derivatives of the glial cells. So when glial cells are growing out embryologically, when they get to the epidermis, they differentiate into Langerhans cells. So those Langerhans cells are connected to the nervous system, which makes that migratory function a little bit questionable in my mind. How is it making its way from the skin of your big toe up to the lymphatic nodes in your popliteal crease in the back of your knee? It doesn't make a whole heck of a lot of sense. It seems like these Langerhans cells, as we're going to describe later, they're communicating through the nervous system or the glial system. We're going to move on. Merkel cells. Merkel cells are sensory receptors. Those are going to detect light touch. Most of the other receptors, not most, all the other receptors, sensory receptors, are all located in this dermal layer. That's the only one that sits in the epidermal layer at the very base, at the basal membrane. Something I want you to note here is the nerve does make synaptic contact with that, but the glial cell does not. Okay, so a little different than the Langerhans cells, and that difference is going to become important down the road. Now, the most important cell for our discussion is the melanocytes. Melanocytes are the ones that are producing our pigment system. They produce those pigments and they transport them through these microtubule system, and they distribute them to all of these different keratinocytes. I guess these guys didn't get any. Now, these melanosomes, which are produced by the melanocyte, they're going to orient themselves around the top of the nucleus, protecting the nucleus from UV light hitting it. I believe there's another function, but we're going to just say that protective function for now. So as we have UV light exposure, these melanocytes are going to produce more pigments. They're going to distribute them more throughout the system. Something else I didn't mention, the crazy shapes of the Langerhans and melanocytes. They're called dendritic cells, meaning they have finger-like projections. I didn't do the best job of representing this, but these guys are squeezing in between those keratinocytes, making their way all around so they have contact with multiple different places. Langerhans cells obviously want to get up to that surface, but the melanocytes want to have the ability to distribute those melanosomes as necessary. Also important to note, this melanocyte has the same structure with a nerve contact to it and glial cell contact to it. So there's that synaptic contact to those melanocytes. Melanocytes also share the same embryologic precursor of a glial cell. So glial cells are what modify to become melanocytes. I think it's very important in my system. This contact, this glial cell precursor derivative, embryologic stem cell line derivative, makes this a system. Meaning I think that these guys, I propose that these melanocytes and Langerhans cells are working with this glial system. I think they're working with the nervous system in some ways too. But as we move further, it's going to make a little more sense. So moving on, structure of a melanosome. So melanosomes are these little structures the melanocytes are producing. These melanosomes obviously contain the melanin pigment. Now, melanin is composed of two different types of pigments. You've got the darker ones, the eumelanins, and then the pheomelanins are the red, yellowish type pigments. Everybody produces different combinations of those. So fairer skinned people are going to have less of the eumelanin, more of the pheomelanin. When we get exposed to UV light, both types of melanins will be produced. So people that are fair skinned will freckle or get a reddish hue, where darker skinned people like myself will get very tan because we're producing so much eumelanin. Everybody, even albinos, still produce melanin pigments. They're just going to have less of them. Albinos are going to have none of the eumelanin. They're going to have small amounts of the pheomelanin. So a melanosome is quite the complex structure. When I started my research on this, I kind of pictured that melanosomes were just organelles filled with pigment, because if you take it for what it's worth, the conventional model is saying those guys are just absorbing UV light, protecting the genetic material. They're a barrier. I don't believe that that is their pure role, because they absorb not only UV light, they also absorb all visible light, red through violet. They even absorb far into that near infrared spectrum. So they're there to take on that charge separation from electromagnetic energy, normal solar radiation. Now, what are they doing with that electromagnetic energy? We talked about charge separation for multiple videos. I went to the detail of how a charge separation occurs in those aromatic proteins, the tyrosine that makes up the melanin pigments. That charge separation is getting transmitted into this complex protein structure. So this center area is composed of what's called a beta-pleated sheet. Protein structures are composed of alpha helixes or beta-pleated sheets. This structure carries electrons in certain manners. So an alpha helix is going to have a chiral flow, meaning that these electrons are only going to flow one direction, but those electrons have to have a matched magnetic spin. Gets a little complicated, we're not going to detail into that, but just keep in mind that alpha helixes are very good at transmitting electrons in one direction. Beta-pleated sheets, much more, how should I say, not understood topic. Research does not quite understand how electron flow through beta-pleated sheets exists. A beta-pleated sheet is going to be composed of protein chains that are interlinked, creating a kinked type of a sheet. In my view, I think these beta-pleated sheets are going to act as semiconductors. All of them are semiconductors, but this semiconductor is going to be set up in such a way that it's going to have a quality to it that is going to contribute to the biophotonic system. So I'll take that slow. We're going to get electromagnetic energy absorbing into these melanin pigments. Those melanin pigments are creating a charge separation that's going to travel through these protein networks. Those protein networks are acting as semiconductors, and in my next lecture we're going to talk about it. Semiconductors have the ability to emit light as electrons are bouncing through the different protein chains. I believe that what's occurring in these pigment systems is biophotonic energy is being created. So we're taking in solar energy and we're converting it to specific biophotonic frequencies. These melanosomes are attached to microtubules. So when you're looking at the structures in these different areas, the keratinocytes have a microtubule system around the nucleus which is holding them in that location. In the melanocytes, you have the microtubules holding those melanosomes over their nucleus too. There's melanosomes scattered throughout the cell. They're not just being transported. Some of them are held in the melanocyte. I think that melanocyte is the location of where photonic absorption is occurring and biophotonic emissions are being created. That information, that energy, is traveling through the microtubule system, and I eventually believe it's going into this glial system, and that glial system is taking in a photonic input of energy, very specific frequency-wise, to the dermatome or the nerves that it's associated with. We're going to elaborate on that quite a bit down the road. With this video, I just want an understanding that photonic energy is being dealt with mostly in the epidermis. It's not getting into these deeper layers. So that indirect effect that's occurring at muscles, ligaments, and tendons that has been shown with red light research, red laser, not light itself, it's happening by an indirect mechanism that I believe begins with the transfer of that energy from solar energy to biophotonic energy, which in my mind is going through this glial cell system. How that happens, that's going to be very complicated. We're going to have to talk about microtubules. We're going to have to talk about semiconduction and how that actually works in proteins. Thank you.